Good morning, children. Welcome again. Today we are going to start the Krebs cycle. In our previous class, we have done glycolysis of respiration, the Krebs cycle or tricarboxylic acid or TCA cycle. Now, the respiratory cycle takes place in mitochondria in the presence of oxygen. It is known as Krebs cycle, and it is given the name after the scientist, hence a Krebs. The cycle is also known as citric acid cycle because of formation of citric acid after the initial product. Now, the steps involved in Krebs cycle are condensation or formation of the citrate. Now, children, in the presence of condensing enzyme citrate synthetase, this acetyl coenzyme A, it reacts with oxaloacetate and it forms 6-carbon compound known as citrate. So, see acetyl coenzyme A in the presence of oxaloacetate and water, and in the presence of citrate synthetase enzyme, it forms citrate and coenzyme. Now, next step is the formation of isocitrate or isomerization. So, in the presence of enzyme aconitase, the citrate undergoes reorganization and they form 6-carbon compound, that is cis-aconitate, and they release water. So, citrate in the presence of aconitase, they form cis-aconitate and water. Then this further reorganized, and in the presence of water and in the presence of enzyme aconitase, it gives isocitrate. Now, the second oxidative decarboxylation takes place. First, it was in the glycolysis. Now, second oxidative decarboxylation takes place in the Krebs cycle, or the formation of alpha glutarate. Now, this oxidative isocitrate, it undergoes oxidative decarboxylation in the presence of enzyme isocitrate dehydrogenase, and oxalosuccinate as is formed as an intermediate which undergoes decarboxylation and form 5-carbon compound, that is known as alpha glutarate, and liberate carbon dioxide. Now, again, third step of oxidative decarboxylation of alpha ketoglutarate takes place. Now, this occurs to form when a 5-carbon compound alpha ketoglutarate, it undergoes dehydrogenation and decarboxylation simultaneously in the presence of enzyme alpha ketoglutarate dehydrogenase complex. It contains TPP, lipoic acid, magnesium, and trans-succinase. So, NAD and coenzyme A are required for this reaction. So, alpha ketoglutarate in the presence of NAD and coenzyme A, and in the presence of enzyme alpha ketoglutarate dehydrogenase, it forms succinyl coenzyme A, NADH, passage, and carbon dioxide is released. Now, synthesis of this ATP or GTP. Now, this succinyl coenzyme A, it is again acted upon by enzyme succinate or succinyl coenzyme A, and they form 4-carbon compound succinate. That the reaction, it releases enough energy to form the ATP or the GTP. Now, this succinyl coenzyme A plus GTP, either ADP, either IDP, and it forms the succinate plus GTP, ATP or ITP. Now, here is the Krebs cycle. Now, this Krebs cycle, as you see, children, so this pyruvic acid which is coming from the glycolysis in the presence of enzyme pyruvate dehydrogenase complex from acetyl coenzyme A, then again it forms the citrate, and aconitase, in the presence of aconitase, it forms a cis-aconitate, and again in the presence of aconitase enzyme, it forms isocitrate, or the isomerization takes place here. Now, in the presence of isocitrate dehydrogenase enzyme, it forms the alpha ketoglutarate. Now, this alpha ketoglutarate the presence of enzyme ketoglutarate dehydrogenase complex forms succinyl coenzyme A. Then again it forms a succinate in the presence of succinyl coenzyme synthetase, and in the presence of succinate dehydrogenase it liberates fumarate. Now fumarate changes into malate, and in the presence of enzyme malate dehydrogenase it changes into oxaloacetate. Now this cycle goes on. So dehydrogenation of oxidation of succinate, this I told you earlier. So 4 carbon compound succinate, it is dehydrogenated to form 4 carbon compound fumarate, and the enzyme succinate dehydrogenase helps in this reaction, and 2 hydrogen atoms are released. Now this is received by FAD, or the flavin adenine dinucleotide, which is reduced to FADH2. Now again this fumarate, or the hydration of fumarate takes place, that is in the presence of water. This fumarate, in the presence of enzyme fumarase, it changes into maltase. Now again oxidation of malate or dehydrogenation takes place here also. Now this malate, it dehydrogenates to form oxaloacetate in the presence of NAD and enzyme malate dehydrogenase. Now this malate, the presence of NAD and enzyme malate dehydrogenase, it forms oxaloacetate and NADH plus H plus. Now this oxaloacetate, it forms at the end of the Krebs cycle, and it recombines with acetyl coenzyme A to start the new Krebs cycle. Now sites of carbon dioxide production, children. Now this carbon dioxide is formed, no carbon dioxide is formed during glycolysis, but 3 molecules of carbon dioxide, they are formed during complete oxidation of 1 pyruvate. So in Rick reaction, so oxidative decarboxylation of pyruvate to acetyl coenzyme A. In Krebs cycle, oxidative decarboxylation of isocitrate, oxidative decarboxylation of alpha ketoglutarate in reaction 4. Now sites of coenzymes reductions. Now during complete oxidation of 1 glucose molecule, the coenzyme, they reduce in the following sites, like the glycolysis and in Krebs cycle. Now what is the significance of this Krebs cycle? Now Krebs cycle, it is a major pathway for the release of energy. So out of total 36 ATP molecules are generated from 1 glucose molecule during respiratory cycle. 30 ATP come from TCA cycle. It means glycolysis seems to provide fuel to the mitochondria. Now Krebs cycle, it provides common pathway for the oxidation of carbohydrates, fatty acids and amino acids. Intermediate products of Krebs cycle, it provides carbon skeleton or raw material for various anabolic pathways like acetyl coenzyme, it used as a raw material for the synthesis of fatty acids. Alpha ketoglutarate acid is used as synthesis of glutamic acid. Succinyl coenzyme A takes part in the synthesis of cytochrome. Oxaloacetic acid is a raw material for the amino acid aspartate. Now here is the difference between the glycolysis and Krebs cycle, children. You will do yourself. Now terminal oxidation we will do in our next class. Till then, thank you and have a nice day.